Hello and welcome to the British Society for Immunology's Connecting on Coronavirus webinar. I'm Jenny Evans, I'm Head of External Affairs at the British Society for Immunology, and I will be your host for today. Thank you all very much for joining us. We're delighted to have you on the line. Thank you too to our supporters of this webinar series, the British Society for Immunology's official journals. Immunology and Clinical and Experimental Immunology are two established journals, and our brand new open access journal, Immunotherapy Advances. Now, as you all know, we here at the British Society for Immunology are very keen to support our members' work on coronavirus and to make sure that the views of the immunology community are well represented in public debate. As part of that, today we've published a briefing note aimed at policymakers on the long-term immunological health effects of COVID-19, which summarises the literature to date and makes research recommendations. Do head over to our website to read a copy of it. It's available at www.immunology.org/coronavirus. Now today, we are very pleased to be joined by one of the members of the expert task force who put together that briefing, Professor Deborah Dunn-Walters. Deborah is a trustee of the British Society for Immunology, and she is Professor of Immunology in the School of Biosciences and Medicine at the University of Surrey. Now today, she's going to talk to us about B cells and antibodies. What does good look like? Now this is obviously a very hot topic that's been discussed lots in the news, so we are all fascinated to hear what Deborah's going to say to us today. I think just to give you a few housekeeping notes, there will be about 30 minutes of presentation with slides, and that will be followed by a 15-minute Q&A at the end. Attendees will be on mute throughout, but please do send in your comments and questions using the chat function. You can do this throughout the webinar, and then we'll try to get through as many of them as we can at the end. We all hope that you enjoy the webinar. Thank you for tuning in. And now, Deborah, I'm going to hand over to you. There we are.
Thank you very much, Jenny. And thank you for inviting me to give this webinar. It was actually a couple of months ago when I agreed to give the webinar and gave the title, and at the time I didn't know the answer to this question. Not that I'm saying I know the answer to the question now, but in just 2 months we know an awful lot more. And I think this is testament to the brilliant immunology community all getting together and working really hard to find out what's going on. I have put references on slides, and my apologies, I don't have time to reference all the papers because there's an immense amount of work has been going on. So why do we want to know about antibodies and antibody tests? And the reason is, the real question is, do we have immunity to the virus? And to measure whether we've got immunity or not is quite complex because there are a lot of different types of cells in the immune system and structures. And so what we're looking for is a proxy that we can measure at scale, because a lot of the measurements for these can't be done at scale, in order to try and get a picture of whether we're immune or not. And B cells, some B cells will turn into plasma cells in this process of immunity, which then secrete antibody, which we can detect by a simple blood test. But there's another reason why we want to know as well, and that's because it's becoming more obvious that SARS-CoV-2 might be the infection agent that gives us the infection, but some of the pathology of the disease is down to perhaps an over-response of the immune system in responding to the virus. And so we need to understand what's happening during the disease. And we need to, the questions we have are what levels of which types of antibody are needed for immunity, where is this antibody, and the dynamics, the timing of the antibody, when does it appear and disappear, and if any, which antibodies might be bad for you. And in order to try and figure out what the differences between these antibodies are, sometimes we want to know which type of B cells make the antibodies. So together with our collaborators at King's, Franco Fratinali's group, we've been working on separating different types of memory B cells using the transcriptome of the cells. So we look at individual cells and see which genes they're expressing. And here we see that we have at least 8 different kinds of memory B cells, in addition to having naive B cells and transitional B cells. And I'll come back to these later. So antibody levels after infection, we're used to thinking that they last forever. So you would only ever get one measles vaccine because the half-life of the antibody is so long that you wouldn't need another one. But then for some of them, we do know that we need a booster every so often because perhaps they would only last, the half-life of the antibody would only be about 10, 11 years. But this is a new virus, and we don't know how long antibody against this virus might last. So there's a really good review here by Paul Kellum and Wendy Barclay, who were looking at all the relatives of this virus and what we know about the immunology there to try and estimate what we would expect for length of immunity against this virus. And estimates at the moment. And in all the papers, and there are a lot of them, there are so many different things you can measure in so many different ways. And human beings have 9 different types of antibody. And some papers will measure IgM. A lot of papers measure IgM as a whole. A few papers will measure individual types of sorry, I think I just said IgM, IgG. They would measure IgG as a whole, whereas individual types of IgG exist that have different structures and perhaps different functions. And there are a few papers that will measure the different types, the subclasses of these antibodies. Similarly, with IgA, we have 2 kinds of IgA in humans, and more often than not, they're measured together and just called IgA. And then IgM is also measured. And when it comes to the virus, what are we looking for in the virus? And this paper here, they actually took lots and lots of different viral proteins and put them out on an array. And then when you test this array, there's healthy serum here, asymptomatic COVID infection here, and some bright spots occurring. So a lot of antibodies in our serum against the nuclear protein here. Now, this is a protein from inside the virus. So you would only really see this when the virus has broken apart. This bright spot here is antibodies against spike protein. Now, the spike protein is all over the outside of the virus. It's the protein that interacts with receptors on the cells in order to infect the cells. So antibodies to here can block infection. Also, with the spike protein, if you model it, the part of the spike protein that interacts with cells is known as the receptor binding domain. And so some people use receptor binding domain in order to measure the antibodies. Now, this paper here looked using IgG, IgA, and IgM at lots of different parts of nuclear protein and listed different parts of the spike protein. And the lowest common denominator here shows that you need the whole nuclear protein for a good reaction here, the whole spike protein, or the receptor binding domain here. So the lowest common denominator are these 3 types of antigens. And these are what most of the papers concentrate on. So when do we get our antibodies? Certainly not right at the start. They're very low antibodies when you first get symptoms, but they do start rapidly increasing so that by 2 to 3 weeks, you've probably peaked in the number of antibodies that you've got against the virus. Now, the IgM and the IgA antibodies then start dropping off after that, whereas the IgG antibodies do stay around for longer. So this paper was a 40-day follow-up time. But they all start increasing at about the same time. This paper was slightly later paper, followed up 70 days after infection. And again, you can see that 2 to 3 weeks is time to get your most antibody. But they all do look as if they're dropping off. When you put the lines of best fit on here, they all have this downward trajectory. In the case of IgM and IgA, to the extent that by 70 days, you perhaps wouldn't expect to see any of that antibody anymore. But for IgG, it would carry on for longer. So this group started trying to extrapolate the data to estimate how long these antibodies would last. And it's quite disappointing, and I think this is why it's hit the news quite a lot. that although you can pick a really good antigen, in this case, it seems that S antigen is better than RBD and N. The half-life of the antibody, median half-life here is only 50 days, and here only maybe 85, 90 days. It's very small. So that's quite disappointing. But there's another group that took a much more optimistic view of these things. And they pointed out that if you take all your data from your patients as a whole, that you might see a decline in antibody levels. But if you then divide your patients up into those that had high titers to begin with, you see that decreasing with time. But if you had low titers to begin with, in some cases, you might almost see an increase with time. So it could depend on the starting point of how much antibody you had in the beginning as to whether you're actually losing it or not as you go along. There are also questions of methodology. There are a lot of different methods and ways of measuring antibody. And scientists will always say, in our hands, we get this. And that basically means that, you know, my water bath might be slightly different than someone else's water bath. And we don't necessarily know that the thermostat's wonky, but we do know that we're getting slightly different results to other people. So we say, well, in our hands, we get this. In this paper, they're very optimistic as well. They see IgG carrying on for a long time. Although when these lines of fit are put in, you see that as you get less data, the areas of uncertainty increase. So this grayed area here shows you how much uncertainty there is about where exactly that line's going. But again, that's for anti-IgG for the spike protein. You perhaps get slightly different results, certainly for IgA and IgM, and perhaps for different antigens when you're looking at the method. So one thing that I particularly took note of for when we do some of these experiments in the future is that we have to inactivate our serum. Immunologists generally don't have category 3 level security laboratories. So for health and safety's sake, you have to inactivate your serum to make sure there's no virus in the serum before you do the tests. And quite common is to use heat inactivation. Now, in this paper, they did use what I think perhaps might have been too much heat. They compared with detergent inactivation or not, but you can see that sometimes something you take for granted in your method might actually be affecting the result. And that's a point to remember in future. This paper, I think, gives me a lot of hope and is one of the best reasons for my optimism. It's the longest follow-up that I've seen, 168 days. Now, if you just look at the 100 days, a lot of papers have followed up to 100 days, and you look at the curve that you get, when you extrapolate from that, it does look like it's going to go down. However, what actually happened was it leveled off. Again, of course, there's areas of uncertainty as you get less data here, but it does look like it's leveling off. The other thing is, up to now, I've just been talking about the level of antibodies, not what the antibodies actually do. Just because we have a high level antibodies doesn't mean that they're all necessarily useful. And one of the most useful things about an antibody is its ability to neutralize the virus. So it will bind to the outside of the virus and stop the virus getting inside a cell. And this paper followed up for 26 weeks, and it still finds quite a number of people have neutralizing titers against the virus. So a lot of other papers have looked at neutralizing ability, and again, different times of follow-up. So we don't know here what's happening after 80, 90 days, but you do generally see decreases. You know, there's no getting away from that fact that in terms of neutralizing ability as well as levels of antibody, there are decreases. And neutralizing ability correlates very well with the levels of antibody, IgG antibody against the S antigen, as you can see here. Perhaps slightly less well when you use the RBD antigen, and definitely less well when you use the nuclear protein antigen. But again, are we looking at a question of if you had a high level of neutralizing ability to begin with, you're going to see a bigger drop. So these are hospitalized patients who have high levels of neutralizing activity here at 30 days, and this is dropping off. Whereas the patients who were asymptomatic or had symptoms, but they were mild, they weren't in hospital, you don't actually see any difference here. Of course, there are individual cases here, and that gets masked in this data. If you look at all the individual cases, there is a drop for all these, even the mild cases, apart from perhaps one there. So there are drops up to 100 days, and we're unsure about what's happening after, say, 100 days. Now, neutralization isn't the only function of antibody. So you can have a neutralizing antibody blocking from going inside the cell, but there's all these other functions, and I'm afraid immunology is known for a number of acronyms and strange numbers. These all begin AD, meaning antibody dependent, and then you have cellular cytotoxicity, neutrophil phagocytosis, etc. And what this is that this region of the antibody is the Fc region. The arms of the antibody at the ends here, they're the bits that are going to bind to the virus, to the antigens. And this part of the antibody is the Fc region, and it interacts with Fc receptors on the surface of cells that mediate these functions like cytotoxicity and phagocytosis, intracellular neutralization, there's receptors inside cells. They also mediate complement deposition, antibody-dependent enhancement. I'm going to come back and talk about this later. That's also dependent on this region. Feedback inhibition of response, because once you've made an immune response, you want to be able to calm it down again later once you've got rid of the virus, so that you're ready for any future challenges that you might have. And this is where I come on to some really nice what we call system serology papers. So not only do they measure IgG and IgA, but they measure different subclasses of IgG. They measure neutralization, they measure binding to Fc receptors, they measure a number of different functions. And in this case, they were looking at DNA vaccines and how well they protected rhesus macaques against future challenge with the virus. And the vaccines weren't completely protective. There were different levels of protection, so they could work out what correlates of protection were in terms of all these different things that they could measure, function and level and type of antibody. And quite nicely, they show that neutralization is important in protecting against infection, but so is complement deposition and phagocytosis. And I recommend this paper to everybody. There's a lot more data here than I could possibly show. But they took serum from patients when they entered into hospital. So at an early stage, they had serum from patients. And they went off and started doing all their tests, their functional tests, looking at the subclasses of antibody, what proteins they were binding to, what antigens they were binding to. And then they got the data back from the hospital to see whether people survived the virus or people didn't survive the virus. And the survivors, they found correlations with IgM against the S protein and with IgA1 against the S protein. So these factors were predictive of survival. Whereas the people who didn't survive, they found that there were correlations with antibodies against the N protein and complement deposition. So I definitely recommend you have a look at that paper. There's a lot more data in there than I can show. And just to put the cat amongst the pigeons a little bit, there was a paper last on a preprint on medRxiv, and they did, again, nice system serology where they look at lots of different functions. In this paper, they also did nasal washes, and this had me rushing off to find out how we do nasal washes. Because what they found was that neutralization correlated with all sorts of different factors related to IgG and IgM when you look in the serum. But when you look in the nasal washes, the antibodies that are protecting the nose, the neutralization and some of these functions correlated with IgA. So again, something else to look at going forward in the follow-up data. And a really interesting thing to me, although it does make me think that I need to go back and revise all my biochemistry from years ago, is that IgG antibodies have sugars attached to them. They're not just all protein. So there's an N-linked glycosylation site here with a sugar attached, and this sugar can have a fucose on it or not. And in these normal patients here in the orange, or normal volunteers, I should say, this is kind of the levels, the relative abundance of fucosylation that you see in IgG1 genes. In COVID patients who were children, it's not too dissimilar. In fact, if anything, they perhaps have higher levels here. But in COVID patients who were adults, you had a decrease in fucosylation that was quite significant, and more so than these other controls that they used, where this is the level of fucosylation that they saw in patients who'd been exposed to influenza. And the reason I'm saying this, and this is relevant, is because the less of this fucose you have on here, the higher the affinity for the Fc receptors are on the cells. So the more potential for some of these activation, some of these effector effects that the antibody might have. So I'll stop talking about antibody for a bit and move on to B cells. What types of B cells might be involved in the disease? And so this is my kind of B cell roadmap. B cells are formed in the bone marrow. They come out as transitional cells and enter a big naive cell pool here, which is hugely diverse, so that when a virus comes along, the chances of one of these recognizing it are really high. And the B cells that recognize the virus will start expanding. And then what we teach everybody in immunology classes is that we have a T-dependent B cell response that goes through a germinal center reaction. So this requires help from other cells. So the B cells require help from T cells, follicular dendritic cells, and a germinal center is formed. And this is a magical place where the B cells can change their receptors and the antibodies that they make by mutating the genes for the antibodies, and then the ones with the best fit to the antigen, to the virus that's attacking you, are selected out. And so it's like a mini Darwinian evolution of B cells going on in the tissues in our body. It takes time. It takes time to make the germinal center, time to go through this process. But what you get at the end of the day coming out of here are some good memory cells with some really nice, highly targeting antibodies. And you get plasma cells that are going to produce these antibodies, and these are quite long-lived. However, there's a shortcut that you can take. So you can get a super fast response coming straight out and going straight to plasma cells without passing the germinal center. And you can have class switching in here as well. And we're calling this, I'm calling this here the extrafollicular response, because these happen in the follicles and these don't go through this lengthy process. So to try and explain a little bit about why I think this is important. If you imagine that you're in a germinal center making an antibody and you're making a really good antibody, this is going to take time. And I couldn't make people, but you get a really good antibody, highly effective, highly targeted. If you're just going to rush, if anybody that can see the target is going to shoot at the target and all rush up, you know, we can all string a bit of string on a piece of wood and try and shoot an arrow. So we can get a huge fast response, but might not necessarily be as effective and as targeted. And this, I think, is really important. And so the way I've drawn these myself, this is a hypothesis. You can tell it's not real data because I'm not very good at drawing curves. And so if you think about a really fast extrafollicular response, and the plasma cells are short-lived because, you know, all these ones here that are going off target, we don't want them to live a long time. So it's controlled by the fact that it's short-lived. So it's really quick, but then it falls off again quite rapidly later. On the other side, you could have this kind of response going on where it takes a while. It's going to be here before you've even made the germinal centers to actually start making these antibodies. So it's quite a slow process, but you're making some really good high affinity switched antibodies here. And if you add these two sets of data together and plot them, you get this. And perhaps this is what we're actually looking at when we're looking in the serum of people to see what levels of antibodies they've got. And maybe it doesn't matter if these have gone, as long as we've got these. So can we actually tell if antibodies are from the germinal center or from the extrafollicular response just by looking at the types of B cells that change? So I showed you this before. This is work we've done. Normally, we use analysis using markers on the surface of cells to separate our cells out and look at different populations of B cells. So we've got a large population in a normal person of naive B cells and transitional cells. And then we've got a reasonable sized population of good classical memory cells that we think might have gone through a germinal center. We have some other populations here. These were unswitched memory B cells, and then we have these B cells here, which don't have this memory marker on, but some of them are switched as well. And so we think that the high affinity, highly targeted antibodies would be made by these populations. And the extrafollicular response would be by the double negative populations, and in particular, this what's labeled here as DN2. So when you look at papers, they will say that in COVID disease, you have an increase in plasma cells. And in severe disease, you definitely get a lot of plasma cells and you see a lot of antibody. And B cells themselves decrease somewhat in the same way that T cells will decrease. When you look in more detail, the IgM memory cells are decreasing, and maybe some of the more classical memory cells are decreasing. But this population that I said was DN2 is increasing quite a lot. And there's also an increase in transitional. This is a really good paper, and this is the increase in transitional B cells seen here. And this is in the mild group. This group of COVID patients had milder disease, where you see this increase in transitional cells. In the group of patients with severe disease, you see an increase here in your plasma cells and in the extrafollicular response cells. This warrants further investigation here, because the transitional B cells, they're B cells, it's a population that probably has definitely has more than one type of B cell in it. You have B cells that are on their way to becoming plasma cells coming out of the bone marrow, but you also have B cells that are regulatory B cells, and their job is to tone down an immune response. And so it might be quite relevant, and we definitely need to investigate this further. So another thing, and this is closer to my area of research, is to look at how diverse the response to something is by sequencing at quite a high level all the different genes that code for the antibodies in your repertoire. And this paper here, this is data where they sequence the antibody repertoire from healthy controls, moderate and severe patients, and where they're illustrating here how the diversity of the response changes. They grouped all their sequences into clones so that you can see whether some are expanded in the repertoire or not, and with the assumption that if it's expanded, it's reacting to the challenge, to the virus. And in healthy controls, that you can see that there's low levels of expansion. Sorry, you can't read these axes. There's low levels of expansion cases. But in some of these severe cases, you have very, very high levels of expansion of just a few different B cells, plasma cells. To the extent that in a couple of these here, where I in a different lab after asking different questions, you know, this could look as if it was a myeloma almost. It's so focused on just single responses here. So you could almost say here, I would anyway, you're putting all your eggs in one basket in this situation. Now, I'm not sure at what time point they looked in this. In the next slide, I'll discuss the time course a bit more. But just to mention that the CDR3 length of these antibodies. The CDR3 is an area of antibodies that encodes the part of the antibody that recognizes the antigen. And it's very, very highly variable. We use that in order to determine what the different clones are. And we've found in the past that as you move from the bone marrow to naive cells to memory cells, you get a decrease in CDR3 length. So as you're maturing your B cell repertoire even more to be able to bind to antigens, you're finding that you're losing antibodies that use a long CDR3 length out of the repertoire. So short CDR3 length is representative of a more matured memory cell. And what they're seeing here is that they've got long CDR3 lengths in their severe cases. And this paper also, and with apologies to the authors for chopping their figures around, but they just had so much data here. But this is really nice data. And they also saw longer CDR3 lengths with the disease. But then here, they also saw lower levels of somatic hypermutation. And this is what happens in a germinal center in a nice, highly focused response. And you can see here in normal people, these are probably largely naive cells. And they're green because they don't have a lot of somatic hypermutation. These are memory cells. They're also bigger dots because there's more clonal expansion there. And in this particular patient, they've got 4 different time points. And you can see at day 8, it doesn't look too much different than the healthy control here, perhaps a bit more clonal expansion. But at day 11, something's happened, and there's been a large influx of lots of new B cells into this repertoire. And it's looking a bit greener. So some of these don't have a lot of somatic hypermutation and perhaps have taken the shortcut without going through the germinal center. And so when you go to day 15, you then see that some of these green ones with not a lot of hypermutation are actually expanding and starting to dominate the repertoire. And even over here, there are expansions here, which I would interpret as being perhaps IgM memory expansions at the expense of a nice, diverse, naive repertoire. So just to reiterate, I think that there are perhaps something that you could consider to be extrafollicular response. It hasn't had a lot of selection to bring down the length of the CDR3 regions and add somatic hypermutation. So there's a polyclonal expansions of cells entering at this stage. So just to move on, sorry, I'm going over slightly, but I'm very close to the end, is that there are severe disease, you get more antibodies. And it's not a function of time since symptom onset. It might be a function of viral load. And the question I have on this one is, when you correlate one thing with another, you have to try and be open to that it could be interpreted either way. It could be that you have a lot of antibodies because they're fighting the disease and antibodies are good. It could be that you have disease because you have a lot of antibodies and antibodies are bad. Now, I don't believe the latter is true, but we do have to watch out for these things. It's very important for safety reasons that we keep watch out. And the things we have to watch out for are antibody-dependent enhancement of infection. The antibody binds to the antigen and it gets taken up into the cell better. And if the virus can then reproduce inside the cell, then you've got enhancement of infection. We don't have any because of the FC receptor function, you can get an increase of inflammation because of antibodies. Now, all these cytokines here have a very good function in the immune system, but just because it's a good thing doesn't mean you shouldn't have too much of a good thing. And you can see when you put the spike protein and serum in with these macrophages, they do produce these inflammatory cytokines. So that's something else to keep investigating. And the off-target effects of autoreactivity, we have to watch out for that as well. I think if there is any, it'll be very rare. But in this paper, the type of pattern of B cells that they saw increasing in severe disease was similar to the type of pattern that they see in systemic autoimmune disease called lupus. I didn't want to end on a pessimistic note, so I've put this slide in. And this paper shows that we definitely get memory cells, certainly to S and RBD. So we can get memory cells. Even if we don't have antibody, we'll have memory cells, and they're much quicker responding second time round. So there's a lot of reasons to be cheerful. The neutralizing antibodies correlate with protection from lower respiratory disease. Maybe not so much at the moment from upper respiratory disease, but we do know that it's possible to make mucosal neutralizing antibodies to protect the nasal cavity. Dropping antibody levels might just be a fact that you're changing the type of antibody that you have, and may not be something that we should be really miserable about. And we have memory B cells and T cells, and so that's always a good thing. This is my last slide, but it's also the most important slide. There are a lot of people at University of Surrey who rolled the sleeves up and volunteered to start helping with COVID efforts in so many different ways. And you know, Immunology, we were trying to recruit a professor of vaccinology, but all professors of vaccinology are rather busy at the moment. But if anybody's interested, please do speak to me. And so we have all these researchers who've been working on collecting samples with our wonderful colleagues at Frimley Park Hospital. But the real reason I've put this slide up is to thank the patients, not only for helping us in our studies, but for all the thousands of studies across. Know any of this if it wasn't for their help. So thank you very much for listening, and I'm happy to take questions. Apologies for the time difference.
Lovely, and thank you very much, Deborah. That was absolutely fascinating, and a wonderful whistle-stop tour, and I'm sure nobody minds running over a couple of minutes. We've been inundated with questions coming in for you on lots of different topics. So I think to start off with, I think really intriguing what you were saying about antibody quality versus quantity and the sort of trade-off there. And I suppose throughout your talk, you've been sort of giving the sort of your sort of average COVID patient, but we know that all patients are individuals, and there's a lot of variety in immune response. I mean, particularly for example, with aging. And I wondered if you could say something about how sort of potentially aging could feed into this.
So we know for certain that aging is very important in this disease. When you look at the data of who gets most severe disease, it's the older people. Who are most likely to catch, it's the older people. Who are least likely to be asymptomatic, it's the older people. Who are most likely to not survive, it's the older people. So it's an incredibly important of different people who have worked on immunology of aging in the past. And at the moment, we don't have any answers as to how aging and this disease, what the causative factors are. But we have lots of clues and hypotheses about where to look. So for example, in the B cells, in aging, I've seen a decrease in IgM memory cells. And I think these are important for all sorts of reasons, but it's interesting that we also see a decrease of these cells in severe COVID disease. Similarly, with aging, you see an increase in double negative cells, and we also see that increase in older people. So maybe the types of B cell response that you get in older people are changing. But these are lots of hypotheses that we need to investigate. It might be that looking at B cells, the reason that things change could be because of lack of T cell help for the B cells. We know that T cells change with age, and you get a decreased number of T cells. We know that the repertoire of antibodies that we have as we get older decreases somewhat as well. So perhaps there's not as much choice around of B cells in order to be able to react to the virus when it comes in. So it might take slightly longer to find the right B cell before they can act. What else haven't I mentioned? Inflammaging. So inflammation is very important in COVID, and we know that some of the cytokines that are increased in COVID have also been known to be increased in older people anyway. So TNF and IL-6, for example. So there's lots of places where we've got good ideas, but what we need to do is to go into the data and then start stratifying. There's so many different levels of data, and what we need to do is try and tease out the difference between severe disease and aging, and take away some of the perhaps underlying conditions that might be confounding the data, and investigate. I don't know if I've forgotten anything. Oh, fucosylation that I mentioned. I think that changes with age as well. So there's an interesting thing that I know nothing about, but which is very interesting to follow up.
Oh, lots and lots of lines for future inquiry, it sounds like.
Yeah.
Oh, and I think we've lost Deborah for good there, I'm afraid. Tell you what, we've had so many questions come in. What we are going to do, we're going to go through them, and we will work with Deborah to put up a blog to answer some more of your questions afterwards. Thank you all very much for tuning in. You will get a feedback survey shortly after this, immediately after the end of this webinar. Please do fill that out and give us your views. I'm really sorry that the cameras and our internet connection has let us down today, but we will be coming to you with more Connecting on Coronavirus webinars shortly. So check out the BSI website for more details. Thank you ever so much for tuning in again, and we'll get that question and answer blog up online as soon as possible. Thank you ever so much, and goodbye.